All right. I do my best to say, oh, yeah, thank you. Oh, dear. That's okay. That's okay. Oh, my gosh. Oh, 5 a.m., you poor baby. Okay. Any questions? What I have is chapter 3. So chapters 1 and 2, when will the test be graded? You're talking about the test, the lecture tests. I go in every day and I try to grade whoever has taken the test. So I will go in tonight before I turn in, and I will grade him tonight. So whoever took the test today, I will grade it tonight. Yeah. Oh, the labs. Okay, the labs take a lot longer to grade. So what I'm doing is I'm grading them little by little. I graded 10 so far. The exam one is this Saturday. Oh, so yes, I know. The exam one, the due date is on Saturday. But if you take the test before that, yeah, I'll go ahead and grade it. The labs are a different story. The labs do take some time to grade. I try to do chunks every day. I do have 2 classes, so far I do a chunk in one class and then another chunk in the other class. I start from the top. So the people with the A last names and B last names are graded. I'm moving my way down. Next time I'll start from the bottom, so I don't discriminate from against the people who have the W's and T's last name. So anyway, but be patient with the lab. Okay, good. So be patient, be a little bit patient with the labs, and obviously over the weekend I'll wrap it up. I know I said 2 or 3 days. I think this is my 2nd day. I don't remember now. It was doing the 5th. Was it doing the 5th? Oh, this is the 5th day. Oh, I'm bad. Yeah. It's gonna get graded by tomorrow. Tomorrow it's free day for me. All I do is grade, so it'll be graded by tomorrow. Yeah. Especially being the first lab, I want it graded ASAP. That way you guys have a sense of how this is graded. So by tomorrow we'll have all the labs graded. And then remember, lab 2 is due Saturday. Yeah. Any other question? Okay, so I do have the PowerPoints already loaded. They should have been loaded. Okay, so I have PowerPoint, the first set of lab 3 PowerPoints. And now, the only thing I'm not too happy about is the way that lab and lecture are not coinciding as much as I would like them to. Chapter 3 deals with microscopy and staining initially, and then we're going to go into the cells. But staining is lab report 4, whereas now we're turning in lab report 2, we're working on 3, and then we'll do 4. But just know that this staining part is going to be seen again in the lab, in lab report 4. So I guess this is a good introduction. I wish they were closer together, but okay. So part of the reason why when we look at microorganisms under the microscope, they have to be stained is because microorganisms, cells, don't have a color. They're translucent. And microscopes have a lit background. So if you're trying to see something that's translucent against a lit background, you don't have contrast. It's very hard to see the microorganism. You have to stain it. The problem with the staining is that it kills the microorganism. So we're looking at dead cells, which is, that's just the way it is. If we use only one dye to stain, we call that a simple stain. And we don't do simple staining in microbiology very often because they're not very useful. In a laboratory, you will take a slide, you will put the organism in the middle of the slide, let it dry. We heat fix it by passing it through a flame. We'll talk about heat fixing. That means that we not only kill the organism, but we glue it to the slide. And then we put a dye in, wash the dye, look at the organism under the microscope. That's a simple stain, which doesn't give you much information. Dyes can be positively charged or negatively charged. Dyes that have a basic pH are positively charged. Dyes that have an acidic pH are negatively charged. Now, this is the deal. Living cells are negatively charged. So that if you use a basic dye, the basic dye will be attracted by the negative charges of the cell, and the cell will get colored. On the other hand, if you use an acidic dye, the acidic dye is negatively charged, just like the cell. So that dye is not going to go into the cell. We use acidic dyes to dye the background, so that we dye everything has a color except the cell. That's called negative staining. And most of the time, we're going to be using basic dyes, however. So notice that when we're trying to make a smear to stain and look at microorganism under the microscope, we have to fix it. We fix it in order to kill the specimen and glue it onto the slide. So kill and stick. Again, we have the acidic dyes, basic dyes, know which one is negatively charged, which one is positively charged. These are examples of basic dyes, which we use in microbiology. These are some examples of acidic dyes. Okay, so a simple stain only allows you to see the bacteria, see what shape it has, what arrangement, but that's really the only information that it gives you. A negative staining technique is one that uses acidic dyes, and in that case, we're going to stain the background. So again, simple stains are not very useful in microbiology. What is really useful in microbiology are what we call differential stains. Differential stains are going to use two dyes instead of one. And in addition to staining the specimen, they're going to put the bacteria into groups, allowing us to identify and classify while staining. So it accomplishes two things. We're able to see the bacteria, the shape, the arrangement of the cells, plus we're going to be able to put the bacteria into groups and say, okay, this organism belongs in this group or in that group, and that allows us it helps us identify it. Okay, so it's yes, we're going to explain how we do that. Absolutely, that's part of what we're going to explain. How this happens. We're going to explain how this happens. So the most important differential stain in microbiology is called the Gram stain. Acid fast stain is another differential stain we're going to talk about. Differential stains are going to take advantage of different chemistry among groups of microorganisms to allow some stains in and some out. And that's basically how that's going to get done. So the Gram stain is such an important stain because it allows us to put bacteria into one of two groups, what we call the Gram-positive group and what we call the Gram-negative group. We said in chapter 1 that most bacteria have a peptidoglycan cell wall, and that is true, except that the arrangement of the peptidoglycan is different between some bacteria, and some bacteria have what we call a Gram-positive cell wall, which has a different peptidoglycan arrangement from the Gram-negative cell wall. So the difference is going to be due to the cell walls of the organisms. Typically, when we Gram stain, we try to use young cells, which are actively growing and dividing and have healthy cell walls. The technique was developed by a microbiologist called Dr. Graham. Before I move on to the stain itself, let me go ahead and go for this slide right here. All right, this is a little bit of a caricature of what a Gram-positive and a Gram-negative cell wall looks like. The Gram-positive cell wall consists of a single thick layer of peptidoglycan. So there's the purple thing is the peptidoglycan. The red thing is the cell membrane. So this is the cell membrane, which is not the cell wall. From chapter 2, you should know the cell membrane is made of phospholipids. Okay, plus some proteins, but the main thing is the phospholipid. The peptidoglycan, we know, was a carbohydrate with amino acids attached. Okay. Gram-negative cell wall. All right, here's the cell membrane. That doesn't change. Now, notice the peptidoglycan. It is a little thin layer of peptidoglycan. So there's one difference between gram-negative and gram-positive cell wall. It has thin peptidoglycan, and plus it has something else. You can see this wavy thing here. That one is being called an outer lipopolysaccharide. Okay, fine. But we're going to call it for now an outer membrane. We'll give it its real name in the next part of the PowerPoint, outer membrane. The outer membrane is made of lipids. It says here lipopolysaccharides, LPS. So the outer membrane is made of lipids. So this is a lipid outer membrane plus the peptidoglycan. So the gram-negative cell wall has two components. It has the peptidoglycan plus the outer membrane. Both together is the cell wall. The key here, though, is that the gram-negative has lipids in its cell wall. The gram-positive doesn't. The cell wall is made of peptidoglycan, which is not a lipid. It's a carbohydrate with amino acids. That's where the difference is gonna come. So let's say you're going to stain two bacteria. Turns out they're gonna be different. One's gonna be gram-positive, one is gonna be gram-negative. You make your smear, you heat fix this slide, and now in the gram stain, the first dye you're going to add is a dye called crystal violet, which has a violet purple color to it. So you add crystal violet, and both cells, gram-positive and gram-negative cells, will take on the dye. This is a basic dye, positively charged. They're going to take on the dye. So if you were to stop right here and look at your bacteria, you'll be able to see the shape of the bacteria, but you will not be able to tell anything else about it. Whether they have a gram-negative or gram-positive cell wall, you won't be able to tell that. Okay, so we move on. The next step in the gram stain is to add what we call a mordant, which is iodine. Iodine is going to create covalent bonds between the crystal violet and the cell. So now the dye is stuck inside the cell, and it will not be easily removed. And the word here is easily removed. So you're going to need a strong chemical to remove that dye. All right, here comes the important step. The important step is called the decolorizing step. We're going to use alcohol. And lipid substances are dissolved by alcohols. Carbohydrates and peptides are not easily harmed by alcohols, whereas lipids are. If you want a degreaser, are alcohols. They remove lipids. So the decolorizer step is very quick. It's a short decolorizing step that lasts maybe 3-5 seconds. And in that quick decolorizing step, the lipid outer membrane cell wall of the gram-negative is harmed, creating pores through which the crystal violet is going to be removed. So the gram-negative cell is going to be decolorized. It's going to lose the color. Whereas the gram-positive, in the brief decolorization step, was not harmed. So the gram-positive kept the color, the crystal violet, the gram-negative lost the color. If you were to stop right here, you'll be able to see the gram-positive cells very easily. The gram-negative will be harder to see because it will not have a color. So here comes the last step, which is a secondary stain called safranin. Gram-positives already have a stain inside. They're not going to take up another one. Gram-negatives were decolorized, lost the first stain. They will take on the second stain. So by the end of the procedure, if everything went right, the gram-positive cells will be bluish purple. Gram-negative cells will be pinkish red. And that's how we'll be able to tell whether you have a gram-negative bacteria or a gram-positive bacteria. Okay. The procedure is not without its problems. If you leave the decolorizer on for too long, now you're going to decolorize the gram-positive. And now you're going to have a gram-positive without a color that will end up with the safranin, and both cells will be red. You won't be able to tell them apart. On the other hand, if you don't decolorize long enough, now the gram-negative will not lose its color and will end up looking like a gram-positive. Okay, so too long a decolorization step, the gram-positive ends up looking like a gram-negative. Too short a decolorization step, the gram-negative looks like a gram-positive. So you can see how this is the important step right there. Okay. There is nothing magic about the dyes. We could switch them, but if we were to do that, then now the colors will be switched, and it will be very difficult to communicate if all of a sudden now the gram-positives are red, the gram-negatives are purple. Okay, because by standard, we've agreed the crystal violet comes first, the secondary stain is the safranin. Yes, you should know the dyes, both for lecture and dye and lab. You should know the order in which they're used. You should know what the problems could be. If you use old cells instead of young cells, an old cell is more likely to be decolorized. So again, a gram-positive could end up as a gram-negative. You should know which type of cell is decolorized, which one is not decolorized. Okay. So that's the gist of the gram reaction. The gram stain is so important that classification of bacteria proceed according to shape and gram reaction. So you have the gram-positive cocci group, the gram-negative cocci, gram-positive rods, gram-negative rods, gram-positive spirals, gram-negative spirals. So you give the shape of the organism and the gram reaction, and that's a classification group. Okay, we will talk more about the gram-negative and the gram-positive cell wall on the second set of PowerPoints. Okay, let's just explaining everything that's happening. Yeah, if we forget to heat fix, we're going to lose our specimen. If we heat fix for too long, we're going to distort the cells. If we use all cultures, they are decolorized more easily, so the gram-positives will be harmed. Okay. So this is, give me a second, gram stain techniques will not stain. Yeah, there are some things in bacteria that will not get stained with the gram stain, like endospores and flagella. Some bacteria have a waxy cell wall, members of the Mycobacterium genus, which we talked about in chapter 2. All right, so here's some examples of a gram stains. This is a really good example here because you can see both gram reactions. You can see the dark purple circular bacteria, which we call cocci, and then you see the little gram negatives, which are actually rods. See that? It's a little bitty, elongated little rod bacteria. So gram-negative rods, gram-positive cocci. This is a gram reaction from a human specimen, from a biological specimen. So these big red blobs that you see here are white blood cells, human white blood cells. The little purple things that you see, those are the bacteria, which are gram-positive bacteria. This is a gram stain performed in a bacteria that is making endospores. Endospores are not stained, not with a gram stain. So you can see the cell, make it a little bit bigger, make it in blue. First of all, you notice that the cell is a gram-positive. Turns out all endospore formers are gram-positive. The endospore is inside the cell. Make one outside the cell. Eventually, the endospore ruptures the cell and is released. But while it is still inside the cell, it will not be stained. The cell is stained, You can see here how these cells have a little empty space inside. That's the endospore. Once the endospore is released, it will not be stained either. So you can see these empty shells there, which are the endospores that have been released. So endospores cannot be stained. We're going to need to come up with a different kind of procedure to stain endospores. Okay, questions? Okay, so that's the gram stain. Okay, so make sure you understand the gram stain both for lecture and lab. Another differential stain that's very important in microbiology is the acid-fast stain. And I must say this is a very important stain in medical microbiology, because in medical microbiology, we are worried about Mycobacterium tuberculosis, right here. Okay, we're always looking out for that one. So this organism is not easily stained because it has a waxy cell wall. The story with the waxy cell wall is that the organism has a cell membrane. Everybody has a cell membrane. It does have peptidoglycan, and it has a single layer of peptidoglycan. So as far as the organization of the peptidoglycan, these guys are gram-positives. However, in addition to the peptidoglycan, they also have a waxy cell wall. And when the cell is young, the waxy cell wall is very thin. As the cell ages, the wax becomes thicker. It makes more and more wax, and it becomes thicker and thicker and thicker. Okay? So that at that point, that cell is very difficult to stain. It would be helpful if we could tell whether we have a Mycobacterium bacteria or not. So let's say you are in a medical lab and you receive a sputum specimen and you're saying, okay, this patient is suspected of having tuberculosis. Wouldn't it be nice if we could say, yeah, the patient definitely has a Mycobacterium? It could be tuberculosis, or no, the patient definitely doesn't have a Mycobacterium. It could not be tuberculosis. So this is a gram-negative cell wall. This is a gram-positive cell wall, positive, which would have the single layer of peptidoglycan, but no waxy anything. Okay, so those are the three possibilities right there. Again, we are going to have a hard time staining, gram staining a Mycobacterium. So we developed a stain that allows us to stain Mycobacterium. The staining technique is called the acid-fast stain. The Mycobacterium cell is called the cell wall is called the acid-fast cell wall. Everything else is going to be called non-acid-fast. Okay, there is another genus of acid-fast bacteria. It's called Nocardia, but it is not medically important, so we kind of ignore it. All right, so the waxy cell wall turns out is called mycolic acid. And what we're going to do here is to heat the slide, and that's going to melt the wax and allow it to stain inside the acid-fast organisms. Notice that here, if you can, you want to use old cells, because the older the bacteria, the more acid-fast it is. Obviously, if you're staining a biological specimen, you cannot, you just stain whatever you're given. You just hope there are acid old cells in there. Okay, so this is the way we're going to do it. Yeah, these cells grow very slowly. Just as a framework, as a reference, quote unquote non-acid-fast bacteria, normal bacteria, I guess you could call it, will double in numbers every 20 minutes. An acid-fast cell double in numbers every 24 hours to 2 or 3 days, depending on how fast it's growing. So it grows tremendously slow compared to a regular bacteria, non-acid fast. Okay, so we're going to start the same way as we do with any stain. We prepare the smear, we heat fix it, and now what we're going to do is we're going to add the first stain, which is carbofuchsia. which has a fuchsia color to it. And then the slide is heat treated, steamed, while we add the carbofuchsia. So essentially what we do is we have a little beaker with water, and there is a Bunsen burner that is heating the water, so there's steam. And we're going to put this slide on top of the beaker, and the specimen is going to be steamed with the carbofuchsia on top, so that the heat is going to weaken the acid-fast cell wall, if there is an acid-fast cell wall in this specimen, but it's going to weaken it, and it's going to allow the carbofuchsia in. So at this point, what's going to happen is, whether you have an acid-fast or a non-acid-fast stain, all the cells are going to take on the carbofuchsia. Then this slide is taken off the heat, so it's heated for about 5 minutes or so. It's taken off the heat, is washed, and then we're going to decolorize. Now, the minute you take these cells out of the heat, they're going to begin to cool. And if the cell has a waxy cell wall, the waxy cell wall will begin to harden again. So that once the acid-fast cell has cooled, it's not going to let go of the dye, because now it again has a hard wax of a cell wall. We're going to decolorize with something very strong, acid alcohol. And we decolorize for a long time. A gram stain is decolorized for 2-3 seconds. These guys are decolorized for 10 seconds or sometimes more. So it's a long decolorization step. What the long decolorization step is going to do is it's going to remove the carbofuchsia from the non-acid fast cells. In spite of the long decolorization step, the acid fast cell will not lose the carbofuchsia, so it will not be decolorized. At the end of the procedure, now we have acid fast cells are fuchsia, non-acid fast have no color, so now they need a second color. We're going to counterstain with methylene blue, so that the non-acid fast cell will take on the methylene blue, the acid fast will retain the carbofuchsia. So at the end of the procedure, you have non-acid fast cells being blue, acid-fast cells being fuchsia in color. And that's how you can tell them apart. Okay, so we go. So acid-fast is not decolorized, non-acid-fast is decolorized. A positive acid-fast out of a specimen is a presumption of Mycobacterium tuberculosis. So we know we have a mycobacterium, we don't know which kind. It may be tuberculosis, it may not. Now we need to take the specimen into more specific tests that will tell us, yes, it's Mycobacterium tuberculosis, no, it's not. Again, this is from a specimen. You can see the fuchsia of the acid-fast cells versus the blue of the non-acid-fast. And here is the another specimen with a little blue, fuchsia acid-fast. Okay, so there's the acid-fast stain. Any questions about that? Keep in mind, the acid-fast stain is not a gram stain. So you have no information about the gram reaction of the non-acid fast. You cannot take a look at these cells and say, oh yeah, these are gram positives. No, they're not. This is a gram stain. You don't know the gram reaction. Okay. All right. Now, another stain is the endospore stain. And one of these days I need to correct these PowerPoints, and instead of calling them spores, I need to call them endospores, which is more precise. Okay, so the endospores are structures that some bacteria can make that are very dormant cells. Essentially what happens is, if the cell senses that it's going to die, or that there's a lack of food, radiation, high heat, the cell immediately begins to dry out on the inside and stabilizes DNA, and begins to secrete layers and layers and layers of peptidoglycan around the DNA. Very stable peptidoglycan and protein components that create an endospore. Eventually, the endospore ruptures the cell and it's freed. Endospores can survive most anything. It can survive high heat, radiation, lack of water, lack of food. They're dormant cells, and they can survive a lot of environmental stress. Once the conditions become better, the endospore will sporulate back into a living cell. So the endospore is dormant, comes back to being a living cell. Okay, so endospores cannot be gram stained. So again, we need to figure out another way to stain them. So endospore stain, Schaefer-Fulton endospore stain. Again, we're going to use heat. So we're going to make the smear, then put this slide on top of a steam bath, and add the dye, which is malachite green. So the first dye is a green dye. So let's see, we have cells here. Let's say this guy has endospores. That's an endospore. This one doesn't have endospores. So when we add the malachite green, the heat is applied for a long time. It can be applied for as long as 8 minutes, 10 minutes, 12 minutes, a long, long time. The heat, the long steam, is going to weaken the endospore and allow it to take on dyes. So at that point, everything takes on the malachite green. Whether the cell has endospores or not, everything gets color green. It says heat smear for 5 minutes. It's more like 15 minutes. It's a long, long steam. The wash with water is long, 30 seconds. That's what's going to remove the malachite green out of anything that's not an endospore. Once the slide is cool again, the endospores harden again, and they're not going to let go of the dye. Anything that is not an endospore will let go of the malachite green. So if you were to stop at that point, you will see the endospores, but you're not going to see anything else. So after the wash with water for 30 seconds, we're going to counter stain with safranin, which is the same stain that was used in the gram stain. And at that point, anything that's not an endospore will not have a color, and it will take on the safranin. So if the bacteria is not making endospores because it cannot make them or because it doesn't want to make them, you will not see the endospores. All you're going to see are the red vegetative cells. On the other hand, if the bacteria is making endospores, you're going to see some red vegetative cells, and you're going to see some vegetative cells with endospores inside and some endospores by themselves. So this is positive for endospores. This one is negative for endospores. Okay, so that's the procedure. Here's what some endospore-forming bacteria will look like. You have the pink cells without endospores. We call them vegetative cells. And we have the endospores inside. Again, this is an endospore stain. You cannot look at these cells and say, oh, these cells are gram-negative. No, they are not gram-negative. You don't know the gram reaction. This is not a gram stain. Okay? This is an endospore stain. The other thing is, if you don't see endospores, you cannot conclusively say this is not an endospore former. You can say it's not making endospores. As far as you can tell, there are no endospores. Obviously, if you see endospores, then you can say, yes, this organism is an endospore former. It can make endospores. Most bacteria will not be able to make endospores. Now, one thing about endospore formers that we have found is that so far, all the bacteria that we have found that can make endospores have been gram-positives, but not all gram-positives can make endospores. Do you know examples of bacterial genera that can make endospores? Bacillus and Clostridium as two of them. So you could, on the other hand, take a look at these and say, oh, this bacteria is an endospore former. It can make endospores. I would think it's going to be a gram-positive. So I'm going to suspect, more likely, when I make the gram stain, I'm going to end up with a gram-positive. Okay? So that you could say. All right. Questions about the endospore stain? Okay, the last of the stains we're going to talk about is the capsule stain. And capsule stains are very different. These are not really differential stains. This is a negative staining technique. A capsule, let's talk about a capsule. So we said that all bacteria have cell membranes. Most bacteria have a cell wall. Some bacteria have capsules. So a capsule is another covering that some bacteria have outside of the cell wall. The importance of a capsule is that it protects the bacteria from the immune system of a human host or an animal host. So capsules are going to make the organism more virulent because it's going to make it less likely for the immune system to be able to fight the encapsulated bacteria. So it's a protective, advantageous structure for a bacteria. The problem with the capsule is that it's very stable. It's made of stable proteins, stable complex polysaccharides. It is antigenic. It doesn't make bonds with anything. This is very stable, so we cannot stain it. However, we could stain everything else. So what we do with the capsule stain is we use an acidic stain to stain the background. Cells are going to not be stained, so they're going to appear clear. That's called a negative staining technique. So at this point, we don't know whether we have capsules or not. All we can see is the background being stained. Then we use a basic stain to stain the cells. Again, the capsule is not going to be stained. So the capsule will appear as this halo against the dark background. So here's how we did it. India ink is the acidic stain being used in the background. Crystal violet is the basic stain used in the middle. This is the halo. The clear halo, which is the capsule around the bacteria. Okay. So again, have an idea of what capsules are and what techniques are used to stain capsules or to see a capsule. We don't really stain it, we detect it. Flagellas can also be stained, but it's very difficult to do. So just know that they can be stained, but we typically don't do that in a teaching lab because it's very difficult. Now, this is another kind of technique. This is a fluorescent dye technique. Fluorescent dyes are very useful because sometimes we can get them to bind to specific structures in cells. And so we can combine fluorescent dyes with antibodies in a technique called immunofluorescence. And that is an important technique. Let me show you how it works. We use immunofluorescence. This is an expensive technique. So we use it to be able to identify bacteria that are difficult or impossible to grow in the laboratory. So Mycobacterium tuberculosis, very difficult to grow in the laboratory. So we have to identify it directly from the specimen. Treponema pallidum, which is the bacteria that causes syphilis, has never been grown in the laboratory. So we again have to identify it from a specimen. We cannot grow it and then identify it, which is typically what we do. We grow it and then we identify it. So that's when these expensive techniques are used. All right, so Treponema pallidum. Let's say you get a specimen, urogenital discharge, suspecting syphilis. Fluorochromes, which are fluorescent dyes, can be attached to antibodies. In this case, the antibodies are going to be anti-treponemal antibodies, the bacteria that causes syphilis. These antibodies can be bought from manufacturers that inject treponema into animals, make the animal get antibodies, and then get the antibodies from the animal, and they attach the antibodies to the fluorescent dyes, and now we have tagged antibodies. So these are anti-treponemal antibodies. So anti-syphilis, if you will. These can be bought. These come in the test kit. Okay, so there's a specimen, and there's a bacteria in the specimen. You don't know whether it's syphilis bacteria or not. You wash the specimen with the immunofluorescent dye that has the antibodies. If the bacteria in the specimen is Treponema pallidum, the antibodies are going to bind to the bacteria. When you wash this slide, the antibodies are not going to wash away. They're going to stick to the bacteria. And when you look at the bacteria at the specimen under the microscope, you'll be able to see the bacteria. And then you can say, oh, this is definitely Treponema pallidum, because the antibodies tagged it. I'm able to see it, and the antibodies are specific for the syphilis bacteria. On the other hand, if the bacteria is not the syphilis bacteria, the antibodies are not going to bind. So when you wash the slide after adding the antibodies will wash away. And when you look at the specimen under the microscope, you won't see anything. Okay, so you can say, okay, that was not syphilis bacteria in the specimen. Oh, what happens when you try to grow? We have not been able to give the bacteria the conditions that it needs for it to grow. So we have not been able to grow it in the laboratory. It just doesn't grow. So we can put the specimen into medium, in which typically most bacteria grow, and treponema doesn't grow. Keep in mind, it has to grow inside cells. We don't do that very well in the laboratory. Bacteria that infects inside of cells, it requires a specific environment that we're not able to give it. So we have not been able to grow it. Actually, there's a lot of bacteria that we cannot grow in the laboratory, and this is one of them. One of the most relevant medically that we cannot grow in the laboratory. So we cannot replicate the environment from the body, from living cells. I don't know how hard they've tried, because once you have a quick test to identify it, then you kind of stop trying to grow it, unless it's for research purposes. So it's typically grown in animals, live animals. Sometimes in tissue it can be grown, but it's very difficult to in a medical microbiology lab to keep tissue cultures just to grow a bacteria. It's just not practical. So this is just some of the terms that you need to know for this chapter. You're welcome. All right. We have pretty much dealt with all of these. Acid-fast stains of a specimen shows the presence of pink rods, given a presumptive test for Mycobacterium tuberculosis. I guess I should have said fuchsia rods, but okay. So the acid-fast stain is positive. There is there are acid-fast organisms in the specimen. However, when you do the immunofluorescent antibody test, the result is negative. So you do have an acid-fast bacteria, but this specific test for Mycobacterium tuberculosis is telling you that that acid-fast bacteria is not the Mycobacterium tuberculosis. So it could be some other Mycobacterium. Okay. Oops. All right, so that is the first PowerPoint for chapter 3. I'm trying to think. Let me tackle a little bit of the second PowerPoint, and that way next Thursday we can finish 3 and start 4. That way we go happy. That way we don't drag it for too long. And actually, 3 is a long. The second part of 3 is a little bit long, so I wouldn't mind going a little bit into it, just a little bit. I'm just waiting for the PowerPoint to load. Okay. I'm going to stop the